I'd like to introduce our speaker today. Once again, I'm Jackie Peterson, and I'm the program manager for the Genetics and Genomics program here at the Stanford Center for Health Education. I want to thank you all for joining me and our fantastic speaker today. Today, I have Professor Ami Bhatt with me. Ami Bhatt is an associate professor at Stanford University in the departments of medicine and genetics. A physician scientist with a strong interest in microbial genomics and metagenomics, she received her MD and PhD from the University of California, San Francisco. She joined the faculty at Stanford University in 2014. In addition to carrying out research at Stanford University, Professor Bhatt has active collaborations worldwide, including in Nigeria and South Africa. She co-founded Global Oncology and serves as a director for Global Oncology for Stanford's Center for Innovation in Global Health. Her team's research program seeks to illuminate the interplay between the microbial environment and host clinical factors in human diseases. Her laboratory studies strain-level dynamics of the microbiome to understand how microbial genomes change over time and predict the functional output of microbiomes. She is keenly interested in how microbes talk to one another and to host cells, and how to leverage this understanding to improve health and treat diseases. Thank you for being here, and I'll now pass it over to Dr. Bhatt.
Thank you so much for the introduction, and thank you all for taking the time to spend an hour with me today thinking about the microbiome. I want you to take a look at this photomicrograph. What is a photomicrograph? A photomicrograph is basically a microscopic picture that one can take. What do you see? And what do you think you're looking at? Well, for me, what I see is an incredibly complicated set of communications that are going on. This happens to be the cross-section of a colon. The colon is the largest part of our intestine. It's at the very distal part, or the last part of our digestive tract. And what it has is on the right side of the picture in the bottom right-hand corner, these blue stained nuclei. That's actually the host side of the colon, or the colonic tissue itself. This happens to be from a mouse, but a human colon would look very similar. On the top left-hand side of the image, what you see are a lot of little tiny dots and rods and spirals. These are bacteria. These are the bacteria that are absolutely teeming in our intestines. And between the two, you see two greenish layers. They're slightly different from one another. And these are the thick and thin layers of the mucus. This mucus is made by the host and separates bacteria from the host. But as you can tell, if you take a really close look at that mucus, you can see tiny little bacteria that are actually invading that demilitarized zone. So we know that we are covered in microbes, filled with microbes, and that they are incredibly important to our health. And over the course of the next 40 minutes or so, we're going to talk about some of the things that we and others have done to investigate this incredibly interesting relationship. Now, I would argue that microbiome science is actually a really old field, even though many of you may think of it as kind of a Johnny-come-lately. In fact, if you take a look at this text from about 30 before the Common Era, this was written by Marcus Varro, who was writing books on agriculture. He noted that certain minute animals, invisible to the eye, breathe there, and, borne by the air, reach the inside of the body by ways of the mouth and nose, and cause diseases which are difficult to be rid of. This is, I think, actually the first description in the literature that we know of bacteria and viruses. Tiny little animals that we can't see that can move from place to place and when transmitted can cause problems. But it really wasn't until the late 1600s and early 1700s until we really had a better understanding of what these microbes actually were. Pictured on the left here is Antony van Leeuwenhoek. He was a famous draper and nobleman in Europe. And what he was really interested in doing was figuring out the weaves of different cloths. Now, as you know, when you're weaving cloth, the more threads you have in a small area, the better the cloth. And to be able to assess how many threads there were in a given, let's say, square inch of cloth, Antony van Leeuwenhoek had been playing around with trying to develop better and better ways to magnify the drapery. And what he ended up doing was actually developing what we now know of as the light microscope. Now, having this cool tool, he decided to not only look at drapery, but also at other interesting things around town. And so he ended up looking at pond scum. He looked at the gunk that was in between his teeth. Remember that this was in an era before common dental practices. And what he found amazed him. He found all kinds of different organisms, big and small, rod-shaped, circular, spiral. Some were sedentary, some moved. And he described these as tiny little animals again, or animalcules. Pictured on the right are actual drawings that Leeuwenhoek drew of these different bacteria that he was seeing. And I would argue that this was actually one of the first times anybody ever saw a bacterium with their own eyes. Of course, over the subsequent years, especially in the 1800s, people started to be able to culture bacteria. They could get bacteria, put them on these petri dishes, as you see on the left, and grow them up into numbers in the millions and billions and even trillions. They then would take these bacteria, put them on a glass slide, and stain them with different stains. And what you see on the right side of this picture is a gram stain, which is a classic way to describe bacteria. Some of you who are scientifically oriented in the audience may know that we describe bacteria as being staining gram positive or gram negative. And so for a long period of time, this is what bacteriology was all about. We collected samples, we cultured them on special media or these petri dishes. We then put them on slides, stained them, and described how they looked under a microscope. But of course, our ability to enumerate microbes was limited at that time. We didn't know how to grow all bacteria. We didn't know what they like to eat. Some of them are totally interdependent and therefore can't be separated from one another. And some of them don't even stain with these specific stains that we have. And so a huge revolution in the field arrived with the advent of next generation DNA sequencing, where suddenly, instead of describing a bacteria by looking at how it looked in terms of its shape and its staining, we were able to sequence all of its genetic code, or DNA, in A's, C's, T's, and G's, and then describe the reads or the words of DNA that we found in a given sample. In so doing, we were able to not only measure one bacteria at a time that we could isolate and culture, but without having to isolate and culture bacteria at all, we could read all kinds of little words and then assign them to different bacteria. Now, this revolution really only started in real zest about 20 years ago. And at this point, our field is centered around this central type of approach. We collect stool samples from humans or other animals. We can also collect other types of samples where microbes live, such as the skin, the mouth, the nares. We then extract DNA from that sample, and then we analyze the sequencing data that we get, or all of the different DNA words that come out, and we try to ask questions like, who's there in this community? We also want to be able to ask, what are they doing? And what do they make? To analyze these huge amounts of data that we're able to generate now, we obviously need efficient ways to analyze this big data. And for that reason, we and others develop computational pipelines that enable us to turn billions of pieces of information into orderly graphs. So over the course of the past 20 or so years, the microbiome field has learned a lot. We know that we have microbes all over our body. We know we have lots of microbes in our mouth, our skin, the colon, and the vagina, for example. We know that, as pictured here, the bacterial types that are present in these different communities are different. And they're not only different within our bodies, but our mouths are very different from our spouse's mouths or our kids' mouths. And so we know that the microbiome varies a lot from individual to individual. By the numbers, we know that we have approximately 10 trillion microbes in or on our bodies. They can make up to 3% of our body mass, and there are at least as many microbial cells as there are human cells in our body. So I would argue that we're almost equal parts them and us. We also know that there are 10 to 100 times as many microbial genes as there are human genes in a given human, which suggests that if we take a look at this from the standpoint of genetic diversity that is contributed to our bodies, bacteria do a lot more of the heavy lifting than our own cells do. But do these bacteria, viruses, fungi, etc., really make a difference in our health? The answer to that is absolutely. Scores of studies have demonstrated that bacteria that live within the large intestine can do a variety of different things, such as extract nutrients from our food. This is certainly true for us, where it is estimated that about 4% of the nutrients that we absorb into our bodies are actually not digestible initially by us, but rather pre-digested for us by our bacteria. But it's even more true in animals like elephants, which are pictured here. Elephants and other ruminant animals such as cows, as you probably know, are entirely herbivorous, so they eat plants. And plants are really hard to break down. Their fibers are very complex, and mammals don't actually possess a lot of enzymes that enable us to break apart these complex fibers, which are sugars stuck together in a long chain. Elephants actually depend on bacteria that live within their guts to do some of that heavy lifting. And you might ask yourself, well, how did those bacteria get there in the first place? Well, it turns out that baby elephants have to eat the poop of adult elephants within their herd to essentially inoculate their guts with these bacteria. It's been nicely demonstrated that baby elephants that are kept away from other adult elephants actually runt, which means that they don't grow properly, probably because they're not able to extract enough nutrients from their food. Nice research in animals has also demonstrated that you can not only lose weight because you don't have the right bacteria in your gut if you're an elephant, you can also gain weight if you have, let's say, the wrong bacteria in your gut if you're a mouse. In very nice research that was done by Peter Turnbaugh when he was at WashU St. Louis, he and Jeff Gordon demonstrated that if you fed skinny mice poop from fat mice, that was actually enough to make them better at extracting nutrients from their food and make them fatter. Now, of course, this is very compelling. It's been followed up with interesting human studies, and this finding hasn't recapitulated well in humans to date. But it's certainly a nice example of how our bacteria are doing a whole lot more than just hanging on for the ride. Beyond extracting nutrients, the gut microbiome can be incredibly important for regulating metabolism, producing important vitamins like B12 for us, and even regulating immunity. Indeed, we know that over the first approximately 3 years of life, the microbiome and our adaptive immune system, meaning our B cells and our T cells, actually co-develop. And early microbial exposures in kids probably have a large role in shaping the immune systems of these children. This has been well demonstrated in animal models, and we know that animals, for example, that are born germ-free or without any bacteria, actually have a whole host of immunological deficits. Now, your early life isn't the only time where you can affect your microbiome. We know that dietary choices throughout life dramatically impact the gut microbiome, be it from formula versus breast milk. Human breast milk actually contains a large variety of human milk oligosaccharides. Once again, oligosaccharides are like fibers. They're chains of sugars put together. And it turns out that these human milk oligosaccharides are not digestible by you or me. They're actually only digestible by certain bacteria that live in the baby's gut. And so the gut microbiomes of babies that are formula fed are actually quite different from the gut microbiomes of babies that are breastfed. There are also choices that we make throughout life that continue to impact our microbiome. For example, pictured here are onions. Onions or alliums, which is what the name of the family is, contain a huge amount of fructooligosaccharides, again, another type of fiber that can actually change the composition of the microbiome. What we know is that the microbiome changes over time, and that from infancy to childhood to adulthood, when people start to experience diseases, the microbiome can shift. We know that all kinds of things, ranging from solid food feeding to having diseases such as obesity or diabetes, can impact the gut microbiome quite dramatically. And so in our lab, what we are keen to understand is how we might be able to save cancer patient lives by understanding and manipulating the microbiome. In particular, in our lab, we focus on hematopoietic cell transplantation, or HCT. As many of you know, HCT is a therapy for people with refractory or relapsed leukemia and lymphoma. It can be a very effective way to treat very aggressive blood cancers, but it comes at a high price. It's a complicated therapy that involves blasting people with anti-cancer drugs or chemotherapy, plus or minus radiation. We give them so much of these DNA-damaging agents that they're rapidly dividing cells, including all of their progenitor blood cells, which are called hematopoietic cells, actually can't recover well. So what we do is we get hematopoietic stem cells from a donor and introduce them into the patient. Those hematopoietic stem cells will eventually take hold, and they will differentiate into red blood cells, white blood cells, and platelets. Now, while transplant is an incredible therapy that has saved a lot of lives, it turns out that it has a very high mortality rate as well. In fact, about 20 to 30% of patients who receive a hematopoietic cell transplant actually die in the first year after their transplantation. And what was really alarming to me was that only about a third of these patients, 37%, die of a relapse of their original cancer, which means that 63% die of treatment-related complications, of which infection is actually a big part of that pie. Many of these non-cancer outcomes that happen to our transplant patients that cause death are associated with changes in the microbiome, as indicated here by the asterisks. And so the open question that our lab tried to tackle a few years ago was, how does the microbiome impact infectious complications of hematopoietic cell transplantation? It seemed reasonable to think that since we have so many bacteria in our gut, maybe those bacteria were impacting whether or not people got infections after transplant. This work was done as a collaboration between Tessa Anderman, a former immunocompromised host infectious disease fellow in my lab, and Fiona Tamburini, a former graduate student in my lab. And what we focused on was trying to bring precision clinical microbiology to the table in the sequencing era. So this starts with a case. At Stanford, we actually saw a patient who was a 39-year-old woman with a B-cell acute lymphoblastic leukemia. She underwent a transplant and was actually doing quite well. Unfortunately, 37 days after her transplant, she developed fevers and chills, and a blood culture showed the growth of the bacterium Staphylococcus epidermidis. Our question was, where did this infection come from? Tessa and Fiona knew that many gut microbes can cause infections, for example, E. coli. But Staph epi, as the name implies, is actually usually a skin commensal, meaning it lives on the skin of people. However, in looking at gut microbiome data from many of our patients, the two of them had noticed that organisms like Staph epidermidis, while very rare in the gut microbiomes of healthy people like me, can actually be quite prevalent in our transplant patients, probably because they've been exposed to a lot of medications that can alter their microbiome. And so Tessa and Fiona asked the question, is the gut microbiome a reservoir of pathogens in this patient population? meaning do they have bacteria in their colons, as pictured in the top part of the diagram, that can make it into the bloodstream, as pictured in the bottom part of the diagram, and cause an infection. To address this question, Fiona wrote a very straightforward bioinformatic tool to help us analyze all of these data. I won't go into detail here, but I'll just give you the name of it, which is Strain Sifter. And what it does is it enables us to identify whether the bacteria in the bloodstream that we're able to identify is the same as one of the bacteria in the gut microbiome. When we applied this method on our patient who had that bloodstream infection, this is actually what her gut microbiome looked like one day prior to her infection. This is a stacked bar histogram. So on the y-axis is the relative abundance of different bacteria. So a big chunk of section means that there's a lot of that bacteria, and a skinny sliver means that there's less. And what you can see is that Staphylococcus epidermidis, which is in the purple and highlighted by a black box, was actually present in the gut microbiome of this individual prior to her infection. Now you may say, well, we've got our smoking gun. Staph epi was in the gut. That's the same bug that caused her infection. But we can't be so fast to conclude that, because Staph epidermidis is present on almost all of us. And in fact, each one of us harbors many, many, many strains, different strains of Staph epi. And so while we found Staph epi in her gut, it didn't mean that it was the same one that was in her bloodstream. Using Fiona's Strain Sifter tool, however, we're able to identify how closely related organisms are to one another. And for patient 13, which is the patient we're talking about today, what we were able to show was that in the blue box, the stool sample that we had for her was actually indistinguishable from the bloodstream isolate in terms of this bacteria. And this is represented on a phylogenetic tree, which is a way to measure evolutionary distance between organisms. When there's a straight line between them, it basically means that they're identical. Now, we found that not only Staph epi infections could originate in the gut, we found that many other organisms also seem to be present in the gut microbiome prior to patients getting infections. This ranged from organisms like Enterococcus faecium and Klebsiella pneumonia to organisms like E. coli and Strep mitis. Of course, we didn't find this for every organism. For a common bloodstream infection called Staph aureus, we didn't, in this particular study, find any evidence of gut colonization. Taken together, what we were able to find was that about 40% of bloodstream infections in this patient population are traceable to the gut microbiome. Which led us to our next question, which is, if pathogens are present in the gut microbiome of these patients, well, how do they get there in the first place? Where do people catch these pathogenic microbes from? What we know about this patient population is that they're in the hospital for a long time. They're very sick. And so we surmise that perhaps patients were catching these pathogenic microbes either from the hospital setting or from one another. And so we inspected whether patient A, as represented on the left, could transmit their microbes to patient B, as pictured on the right. And if this was the case, might things like being in the same room or the same ward at the same time be risk factors for this? So to ask and address this question, Ben Cyrinoshan, a former graduate student in the lab, teamed up with Hua Tang, a professor of statistics and genetics, and Niaz Vinay, who is the head of our clinical microbiology group. And what they found was actually interesting. We studied hundreds of patients who had been on the same ward over the course of about 15 months. And I'll highlight just a couple of cases that we thought were interesting. First, an overall finding. Most patients don't seem to be catching microbes from their neighbors that much. And so we found evidence of transmission, but it was rare. This is an example of transmission of an Enterococcus faecium, which is a pathogen that can cause bad bloodstream infections, and oftentimes this organism is quite drug resistant. Now on the top part of the diagram, once again, you have a stack bar histogram describing the relative abundance of different bacteria in that top patient's microbiome. And you can see that that top patient was pretty much filled with Enterococcus faecium, as highlighted in blue. If you take a look above that diagram, you can see a skinny bar, and that bar indicates when the top patient actually overlapped with the bottom patient as a roommate or in the hospital at the same time. While you can't see it very well, what we found was that the blue organism that is present in the top individual actually seems to appear in the bottom individual, albeit at a low relative abundance, only 0.4%, shortly after the time period where the two are roommates, suggesting that there was a direct transmission event between the top individual and the bottom individual. Put another way, our bottom patient actually caught Enterococcus faecium from the top individual in their gut microbiome. And there, the gut microbiome E. faecium could lie in wait until there was an opportune time to get into the bloodstream and cause an infection. We not only found this for pathogens like Enterococcus faecium, we actually also found this for good microbes, such as Hungetella hathawayii. Now, Hungetella hathawayii is a commensal organism that is thought to actually be very anti-inflammatory and probably healthy in our guts. And once again, as you can see in this image, we've got one patient on the top, second patient on the bottom. You can see that there was transmission of a Hungetella hathawayii from the top patient to the bottom patient. And that happened shortly after they were roommates. Interestingly, this bottom patient did have a Hungetella hathawayii at the very beginning of this time course. And when we noticed that, we thought, oh, well, maybe these patients just had the same organism to begin with. However, when we compared the Hungetella hathawayii that the bottom patient had at day 0 versus at day 58, we found that those were totally different, meaning that the original Hungetella hathawayii that the bottom patient had been replaced by the one that it got from its roommate. Put together, what we were able to demonstrate was that we could use strain-resolved evidence that gut commensals and pathogens can be transmitted horizontally between adults, meaning that they can get passed from adult to adult in this hospital setting. So if patients can catch pathogens, a question then follows. If I want to prevent infections, and I know that infections in the bloodstream often originate in the gut, and I know that patients can catch these microbes in their gut, how might we manage patient microbiomes to prevent acquisition of these pathogens and eventual outgrowth of them? To answer this question, several members of my lab have worked on a series of projects where we have tried to modify the microbiome using techniques that range from fecal microbiota transplant to antibiotics to prebiotics. So there are 3 main ways that you can change a microbiome in broad brushstrokes. One is to use antibiotics that kill certain bacteria. The next is to use probiotics or live bacterial therapies, a very extreme version of which is fecal microbiota transplantation, which many of you have probably heard of, where you give the feces of one individual to another. The third way that you can change the microbiome, that should come as no surprise to you, given that we talked about it earlier in the seminar, is prebiotics or fibers. It turns out that we can give fibers that selectively feed certain components of our microbes over others. And this can also be a way to remodel the gut microbiome. In our first work in this space, we worked on fecal microbiota transplantation, and this was a collaboration with Libby Homan's group at the Mass General Hospital. While when we do FMT, we don't do what's pictured on the left, which is an image of 2 people's colons stuck together. What we actually do is something more like what's pictured on the right. We take feces, we filter them, we test people extensively to make sure they don't have pathogens that we could transmit, and then we can package them into things like these capsules or so-called crapsules. Patients then take 15 of these pills one day, another 15 of these pills the next day, and that's usually enough to actually colonize their gut microbiomes, at least in the short term, with these microbes. While I won't go into details of the findings that we had in this study, I'll summarize it by saying that we found that the microbes that the donor gives to the recipient through this FMT colonize well in the recipient for the first few weeks, but in the long term, they end up getting washed out by other organisms, suggesting that short-term colonization is actually quite prevalent, but long-term changes in the microbiome are actually hard to achieve with a 2-time fecal microbiota transplant. Another way to change the microbiota is actually to use prebiotics, as we've discussed. And Tessa Anderman, along with Andrew Rezvani, carried out a prebiotic pilot trial where we gave a prebiotic, in this case, fructooligosaccharides, which you'll remember is a fiber that is commonly found in plants such as onions. We gave this fructooligosaccharides to patients undergoing a transplantation. We did a dose escalation single-arm study. 15 patients were enrolled, and the FOS, or fructooligosaccharides, was tolerated well. While this was going on, Tessa collected stool samples and blood samples from these patients, as well as control patients who were not receiving the fructooligosaccharides. While we found no major differences in the gut microbiome between the individuals who got the FOS and people who did not, we did find that fructooligosaccharides were probably important for producing this type of small molecule called butyrate. Fructooligosaccharides, once again, are these complex sugars, and bacteria can metabolize them into tiny little pieces, which are called butyrate, or short-chain fatty acids. And butyrate had been demonstrated by others in the field to actually be very anti-inflammatory. In this particular example, butyrate could ameliorate or improve graft versus host disease, which is an inflammatory disorder. The way butyrate works is it actually can trigger T regulatory cells, which are an inflammation suppressing cell type in the immune system, to proliferate. And so we had surmised that FOS might increase the abundance of Tregs, these anti-inflammatory T cells, in the patients who got it. And indeed, we found, as is outlined in the red box, that patients who got FOS had more FOXP3-CD3 cells, which are T regulatory cells, than control patients. So we've talked about probiotics, or FMT, we've talked about prebiotics. The other arm of this three-legged stool is antibiotics. Now, typically we think of antibiotics as doing harm to the microbiome, but a series of studies done over the past approximately 5 decades have suggested that giving patients non-absorbable antibiotics, meaning antibiotics that can act on the intestine, inside of the intestine, but don't get into our bloodstream, can actually improve the outcomes of patients who are undergoing hematopoietic cell transplantations. So Jen Wangbo, who is a physician scientist in Boston at Boston Children's Hospital, and Chris Severin, who is a pediatric hematologist and oncologist in my lab, teamed up to do a randomized controlled trial of this practice of gut decontamination in pediatric patients undergoing allogeneic hematopoietic cell transplantation. What they did was actually give, once again, some patients the gut decontamination and other patients did not get any, so they were the controls, and they looked for differences in the patient populations. Now, this was a very small study, and while this did not reach statistical significance, we did find that the patients who got gut decontamination had many fewer bloodstream infections than the patients who did not get the antibiotics, which suggests that these gut decontamination antibiotics might be clearing out some of those pathogens that we've demonstrated can be present in the gut microbiomes of these individuals. Chris was further able to demonstrate that in every case in the non-gut decontamination arm, or the controls, where they had an infection, the infection was traceable back to the gut microbiome. Taken together, what we found was that this is days after transplant on the x-axis, and the percent of people who had a bloodstream infection with concomitant colonization of the gut with that organism. we see that there is a big difference, a visible difference, between those individuals who had no gut decontamination versus those who had gut decontamination, which suggests that gut decontamination may decrease the incidence of these bloodstream infections. So coming back to this picture, I hope I've convinced you that we and others have developed exquisitely sensitive methods to figure out who's there in the microbiome. And we know that measuring microbes at the strain level matters because there are millions of different strains of, for example, an E. coli out there in the world. We talked about precision clinical microbiology in the sequencing era. We talked about how we can track microbes between patients. And finally, we talked about how we can modify the microbiome using fecal microbiota transplants, antibiotics, and prebiotics. So that's what we've done. What are we doing next? I thought I would give you a bit of a sneak preview into the work that we're up to now. And as we look to the future, we have become extremely interested in co-opting microbial communication systems so that we can precisely manipulate medicine. When we look at this figure, part of what we're trying to do is really zoom in to the bacterial side of things. And when we zoom in, I imagine a scene like this is going on, where you have wonderful and exciting conversations going on between bacteria. And not only between bacteria, but maybe also even between bacteria and us. This is actually not so far-fetched. We know from previous research that microbes can use small molecules or chemicals to talk within species, so they can talk to their own. They can talk between species and actually also engage in bacterial warfare, killing others off. And they can use these small molecules to even talk between domains of life, meaning between bacteria and humans. This small molecule collection of communication signals has been exploited extensively in what I call nature's medicine cabinet. Many of the drugs that we use in modern medicine today, ranging from the chemotherapy doxorubicin to the topical antimicrobial agent bacitracin, are actually derived from bacteria. But while these small molecules or chemicals have been well mined for their use in precision medicine, proteins have not been so well mined. Proteins, however, are optimal signaling biomolecules. They're diverse, they're very highly specific, they're biocompatible, and we can synthesize them scalably. And so the question that we now have in our lab is, might microbes use small proteins to communicate? A greatly enabling piece of work that has allowed us to peer into this new and exciting realm of microprotein-based communication was carried out by Gila Sparrow, a former postdoc in the lab. Using computational biology approaches and evolutionary theory, she was able to discover thousands of hidden small proteins in microbial genomes. In total, she discovered about 4,500 new families. Most of these are of unknown function, and about 30% of them we predict are involved in cell-cell communication. So as we move forward, what we hope to be able to do is take this blue bacteria on the left, identify small proteins that it makes, like these pink blobs, figure out what host cells or human cells they communicate with, as pictured in the middle, and eventually exploit this information to be able to treat the cancers that are pictured on the right. We'd love to be able to understand how we might use microbial proteins to regulate human biology and to improve cancer treatment to therapy. So taken together, our relationship with our microbes is most certainly complicated. Some of the microbes are bad, some of the microbes are good. Sometimes it just depends on if they're in the wrong place at the wrong time. But we have lots of opportunities now that we have sharp and effective tools to measure microbes to study how they impact host health. One of the most important things that we do in our lab is collaborate broadly across the world, as you heard in the introduction. and really proud and honored to collaborate with the H3 Africa Genomics Consortium to improve the study of microbiomes and genomes all over the world, not just in places where wealthy people live and researchers happen to reside. And so we've been very fortunate to collaborate with Professor Hazelhurst, Boki Oduwaran, and many others in South Africa to actually carry out some of the first comprehensive microbiome analyses of individuals in Africa. And I'm actually very excited because our first major paper on this, or a set of major papers on this, just came out over the course of the last year, with the most recent paper coming out 2 days ago. We're interested in these not only because of diseases like cancer, but as pictured on the previous slide, issues such as obesity and heart disease have become of increasing interest, not only in high-income countries like the United States, but all over the world, including in South Africa. So there, our focus has been on obesity in women and cardiometabolic disorders. Soon, we actually plan to study menopause. As I mentioned before, we're keen to figure out how microbial therapy might boost cancer immunotherapy. And I would be remiss to not mention that we are also very keen to understand the gut-brain axis, focusing on a couple of major diseases, Alzheimer's disease and Parkinson's disease. Finally, I'll just end with a note about how microbial genomes are interesting not only because they tell us how microbes function, but they also are amazing treasure troves of cool enzymes and machinery. I'm guessing that many of you in the audience have heard about CRISPR, which is basically a system. The CRISPR-Cas9 system is a system that exists in bacteria to fight off phages, which are viruses that infect bacteria. This entire enzyme system has been co-opted for genome editing. There are tons of companies in this space, and this type of editing technology has enabled medical practitioners to fix errors in people's genomes to try and reverse diseases. Most recently, we and a set of collaborators in the BASIC, Bintu, and SHU labs have worked together to identify a new class of genome editing enzymes from bacterial genomes. This class had been previously described in a limited way. These are the large serine recombinases. We've been able to discover thousands of additional candidates and identified the specific places where they target in the genome, which might enable us and others to take large amounts of DNA cargo and deliver them to safe places in the human genome, thereby treating people who have complicated gene deletion disorders. So we've talked a lot about a lot of things today, ranging from who's there in our microbiome to how we measure them, how microbes travel between human bodies and within a human body to cause problems. And we've touched on how things are moving in a new direction to try and better understand microbial communication so we can co-opt those machineries to try and improve modern medicine. There is a lot of promise, as I'm sure you can appreciate, and a lot more work to do. I look forward to hearing your questions. And I just want to end by thanking my entire lab. As I mentioned, I'm very fortunate to work with an incredibly talented and passionate group of individuals, our many collaborators, our funding sources, my recent grads, and of course, all the patients and people who have participated in our studies. I look forward to your questions.
Great. Thank you so much, Professor Bhatt, for that presentation. That was really fascinating. So for the first question, if a baby elephant is inoculated by feeding on parents' feces to acquire the microbiomes, what's the mechanism that a human infant uses to acquire microbiomes?
Yeah, great question. So it's been demonstrated that infants actually acquire their first microbes during birth, which means that the microbiomes of infants born by vaginal delivery are actually quite different from the microbiomes of infants that are born by C-section. It's been demonstrated that in the very, very early part of life, infants actually born by vaginal delivery are colonized with a lot of vaginal microbes. And infants born by C-section, it won't surprise you, are colonized with a lot of skin microbes. Over the course of time, however, infants probably continue to collect microbes from their environment, from their mom and dads, from the skins of people around them. A great mentor of mine, Stan Falco, who was a really world famous microbiologist, used to joke that the world is covered in a fecal veneer, meaning that there are microbes everywhere that, are from the gut microbiome. And so there's probably a little bit of that as well. And so as babies actually move on in life, they acquire microbes from their environment. And there's actually a very standard process along which the microbiome builds, which is actually super fascinating and a topic in and of itself.
That's really interesting. Thank you. The next question is, when the microbes that are transferred via FMT may go away over time, is it because the native microbiome has pathogens in it that take over, or the native microbiome just resets?
This is an excellent question that we don't exactly know the answer to yet. Mm-hmm. I would speculate that what happens is there are microbes from the native microbiome, the original recipient microbiome, that are kind of lying in wait. And so they're suppressed to very low levels, and over time they can actually increase in amount. I also think that people reacquire probably their microbes from their environment. I think part of the reason that the donor microbes go away might be due to actual warfare, which is what you have described, right? They're being fought off by the recipient microbiome. Another reason that they might go away is because of lifestyle differences. So for example, I happen to be vegetarian. If I got a fecal microbiota transplant from somebody who ate mostly meat, a lot of their microbes would not find a happy home in me because I wouldn't be feeding them the right foods. And so I think that also plays a role in figuring out which microbes might be able to stay for longer term versus which end up getting kicked out. I also think that the question you asked is very related to the previous question. We talked about this very orderly process of microbiome assembly that happens in infants. And if you think about that really orderly process like a pyramid, if you take out a piece of the middle of the pyramid, it's really hard to replace it with a piece that's a different size or shape. And so I think what happens is our microbes are very dependent on each other in a complex network. And that's probably also why the introduction of a few microbes via FMT or probiotics is unlikely to have long-term effects unless we keep on giving people those microbes.
Great, thank you. The next question is, what are some good ways to improve and protect our gut health and microbiomes?
So there are several things that we can do to try and improve our gut health and protect our microbiomes. I think one thing that is incredibly important is trying to eat a diverse diet. In fact, many different diets have been studied, and many have posited that certain diets are better for the microbiome than others. I think some of the most convincing data on this comes from a study that found that eating more than 30 different types of plants in a week, is one of the biggest predictors of microbiome diversity. And so I think keeping a diverse diet is incredibly important. Staying hydrated is probably an important part of this equation. Many people do want to take probiotics or eat fermented foods. Obviously, many cultures across the world have had long traditions of fermented foods, and I think that it's advisable. However, with probiotics, it's hard to know what you're getting. Probiotics aren't regulated the same way medications that we take are. And so you never know if what's on the label of the bottle is actually what's in the capsule that you're taking. Furthermore, as we mentioned before, it's hard to know if those microbes that you take in probiotics will really stick or not. So I think some of the best things that one can do is try to eat a diverse diet, to try to keep a relative diversity of foods on your plate, to stay well hydrated, and to eat the types of fermented foods that you enjoy.
Great, thank you. Along those lines, can knowledge of individual gut microbes provide a personalized plan for improved diet and healthy lifestyles?
So there are many groups that are actually trying to achieve this objective of taking a look at your microbiome and then trying to comment on what might help you achieve a better microbiome. I look at microbiomes all day long. It's actually my job, and I'm also a physician. And I've got to say, I don't think the science is quite there yet. That said, there are efforts that are underway, both on the research side of the equation and also on the industry side of the equation, to try to help people diversify their microbiomes. I do think as we look to the future, we will be able to better and better understand the microbiome, to understand what our microbes like to eat and think about ways that we can feed them. Sometimes I like to joke that, we're not just eating for ourselves, we're eating for trillions. And so, knowing who's in our gut microbiomes and what they like to eat might help us actually modulate and modify our microbiomes. I can certainly tell you that one of the reasons I think fibers are so important is that simple sugars are actually preferentially metabolized by most pathogens. And so one way that we can suppress the growth of pathogens in the gut microbiome is really to try to limit simple sugars.
Excellent. There is another question. Is there a significant difference in the health of breastfed babies compared to the health of formula-fed babies because of the differences in their microbiomes?
So this has been extensively studied in the medical literature. In the microbiome literature, while there are differences in the microbiomes of breastfed and formula-fed babies early on in life, it's unclear whether those changes actually persist in the long term. And there's obviously the intersection area too. There are a lot of babies that are fed by both breast milk and formula. And so that remains another kind of interesting area for investigation. I think, based on the data that we have right now, the best answer I could probably give is that there are probably no major long-term effects. There may be some subtle ones. And in the setting where there is extended formula feeding before the introduction of solid foods, there may be some consequences in terms of immune maturation. Because remember, those early parts of life are where the microbiome is changing and the immune system is growing. And so they're really influencing one another. I will say one last thought on this is that while in the United States, for example, the practice is not to introduce complementary foods or solid foods until around 6 months of life, that is actually very different in different countries around the world. And so there are countries where solid foods are introduced very, very early on. And so I'd be very curious to know how that impacts the growing gut microbiome of babies as well.
And the next question is, are there any other foods in addition to onions that are beneficial for good bacterial strains?
Absolutely. I think there are basically any fibrous food is a good choice. Jerusalem artichokes have a ton of fiber. Eating fruits with the skins on. So, don't get rid of that apple peel. Just wash your apple and eat it. Leafy greens have a lot of fiber as well. And it's interesting, all of these different foods have different types of fiber. Legumes also have a wide variety of fibers. And so, lentils have different fibers from garbanzo beans, which have different fibers from pinto beans. And so, exploring a variety of these different plant-based foods can be extremely helpful for bringing more fiber into your diet.
And how will cleanliness due to COVID affect the microbiome?
This has been a topic of a lot of conversation. We have kids who have been, in much more hygienic homes, who aren't interacting with other kids, aren't interacting with almost any other adults other than their parents. I think the anticipation is that this will have some impact on the microbiome. If you ask me to guess, I would guess that the impact is going to be relatively small, just because it's so difficult to live a totally clean, sterile life. So long as you're going out there in the world, you're being exposed to tons of bacteria. Places like soil, for example, are just teeming with bacteria, way more microbes in soil per square or cubic inch than in our guts. And so I think it's really difficult to limit our exposures entirely. But I do anticipate that as this is studied in the years to come, there will be subtle changes. And I'm curious to see the impact of this. I think that that'll not only be important on the gut microbiome side of things, but possibly also on the skin microbiome side of things. And so we'll have to wait to see. I do think on balance, though, we're always weighing risks and benefits. And I think during the period of time where there was so much uncertainty, certainly it was worth the potential risk of deficits in the microbiome to avoid the sure risk of potential exposure to COVID for young and unvaccinated children.
Thank you. So do oncology nutritionists recommend butyrate for patients with graft versus host disease?
So butyrate itself isn't recommended, and part of the reason it isn't recommended is it really honestly just stinks. Butyrate, I can't believe I'm going to say this on a webinar, butyrate smells like farts. And so nobody wants to take it, even mice don't want to eat it. And that's part of why this workaround has arisen, which is giving people fiber so that butyrate can be made when the fiber is metabolized. The other challenge is, of course, butyrate is actually a small molecule. If you remember what it looks like, it's tiny short-chain fatty acid. So it can also be absorbed very high in the GI tract, which means that it may not get down to the parts of the intestine where graft versus host disease is important. But absolutely, it has been investigated. You know, people have thought about, are there ways to formulate butyrate into like a super olfactory contained capsule so people can take it. But it hasn't quite hit prime time yet.
Great, thank you. Last question that we have time for today. Since you talked about boosting regulatory T cells, I imagine there have been studies also regarding the microbiome and inflammatory diseases, specifically like ulcerative colitis and Crohn's.
Yeah, so there have been extensive studies on ulcerative colitis and Crohn's disease, which are two different flavors of inflammatory bowel disease. These diseases, for those who aren't familiar, are caused by our immune systems attacking our guts. And the two diseases are slightly different from one another, but actually share some similarities. There have been, like I said, extensive studies in this regard. A lot of people have actually postulated that fecal microbiota transplant might be helpful in changing the microbiomes of these individuals to try and increase regulatory T cell proliferation and suppress inflammation. That's actually some of the work where it's been found that while a fecal microbiota transplantation might help people for a while, the effect seems to go away. And we wonder if our findings that donor strains don't really engraft for the long term might be responsible for this finding. And so there are studies underway with both fecal microbiota transplant and probiotics, live bacterial therapies, to give people those therapies on a routine basis to try and suppress inflammation. So I anticipate that there will continue to be exciting research in this realm. I will say, from a physician perspective, if we could find a way to find the molecules that are actually responsible for modulating the immune system and doing much more than just inducing Treg proliferation, I'd much rather develop those molecules into medications that we can give patients rather than taking the risk of giving somebody a fecal microbiota transplant filled with lots of known and many more unknown bacteria where I could possibly be introducing a pathogen. And so I would say the field is really moving in the direction of thinking about what the molecules are that the microbes use to engage in crosstalk and then leveraging those as potential drug-like compounds.
Perfect. Thank you so much. I think that's all the questions that we have time for today. So we're going to need to wrap up. But if you'd like to learn more from Dr. Bhatt and the rest of the genetics faculty, we encourage you to join our genetics and genomics program. You have more than 90 hours of content to learn from them, as well as access to videos and exercises to sharpen your skills. And a link to the recording of this session will be emailed to you soon, so feel free to revisit this content. Once again, thank you for joining us, and we hope you have a great week. We hope to see you again in future sessions. And thank you so much, Dr. Bhatt, for joining us today. This was a great presentation. We appreciate it.
Thank you very much. Have a great day, everyone.